one that was missed frequently. Let me look at the question here. So for that one, it said, which of the following cells have reduced or very little active telomerase? So the correct answer to that was somatic cells. So once cells differentiate to become body cells, they typically downregulate or turn off altogether telomerase. And then that's what's unique about cancer cells is they reactivate it. So A should have been the correct answer for that one. And then number 17 was also missed. Actually, that was the most missed question on the exam. I think that one was about the order. So that one was for transcription. And so I think the biggest thing that people didn't realize is that the sigma and RNA polymerase come together before they even bind to the DNA. So the correct answer should be C. So first thing that happens is sigma binds to RNA polymerase, and then the sigma part binds to the promoter, but it's the holoenzyme that is recruited. And then the double helix of DNA is unwound. That's where it goes from the closed complex to the open complex. And then transcription begins, and to escape the promoter, you have to let go of sigma factor. So correct answer for that one should have been C. Number 21. Okay, this was one, I think we kind of practiced this, but I think it's just when you come down to it, it's really about making sure that the directionality is correct. So it says if the coding strand of the DNA reads GTA, then what would the anti-codon be? So we're basically going from DNA, we have to figure out what the mRNA would be, and then we can figure out what the tRNA would be. This would be the codon, this would be the anti-codon. So, and then the directionality is important. So if we go from coding to the codon, what would the sequence be for the mRNA? Yeah, GUA. And then we're finding the complement, so CAU, but we have to flip the orientation, and that's, I think, the key part. So it's 3' and 5' because those two are actually physically paired together. And so in the answers, all the answers are written 5' to 3', so I think that's where people got confused. So it should be, let's see which one is UAC, 5' to 3'. So that would be D. But, you know, if you didn't flip it around, then that's where I think the confusion came in. 26 and 29 were the other two. So for 26, this is true false. Ribosome checks the incoming tRNA to ensure that correct amino acid is attached. And so there is no check. So does anybody remember at what point there is some checking? Yeah.
tRNA synthetase.
Yeah. Yep. tRNA synthetase is the thing that checks. But after that, there's no checking. So if it makes a mistake, then it's just going to incorporate the wrong amino acid in, until that tRNA synthetase does its job again. So that should have been false. Okay. And then 29, so gene-specific transcription factors are required to recruit RNA pol-2 to the core promoter. So it's actually just, that should have been general transcription factors for that to be correct. So gene-specific is doing that enhancing, and it's acting through the mediator complex, but it's not actually required. So that one should have been false. Okay, does anybody have any other questions? I know you just got them back, so you can look them over. If you see anything that you think should be correct, or, I'm always happy to hear your reasoning. I did say that I would do this. So I have an exam analysis. The class average was actually pretty good. It was like a 79%. I know you don't think that's high, but for a majors level class, that's pretty good. I do have this if you want to, so entirely optional. But what it is it's kind of looking at your answers and the ones you got wrong and deciding why. And so there's a lot of different options here about, I had trouble interpreting it. I didn't think this was important, so I really didn't, prepare that material or study that material. So what you do is for every single one that you got wrong, you write the question, you classify the reason, and then the correction is going back and finding or giving a reason for why the correct answer is correct. So if you only missed 4, then you only have to do that for 4, but you have to do it for all the ones that you missed. The last part of it is writing, kind of reflecting on your study, how you study, and then just trying to think about what you could do differently. I mean, in every class you always have to figure out what's the best way to study for this type of material. So if you want to do that, you can. It's worth 5 points added onto your test. So that's a half a letter grade, but you don't have to do that if you don't want to. So I'll just leave it up here. If anybody does want to at some point pick that up, you can pick that up. Okay, and then the other thing that I posted last night, so I wanted to show you where that is. It feels so hot today.
It's like muggy in here.
What did you say?
It's like muggy inside.
Yeah. Feels extremely muggy. Okay. I think I have. Actually, let me use this. All right, so I posted a study guide for our test on Friday, especially because the lab material, there was like just so much information that you could know. So I just wanted to kind of narrow it down to what you should focus on. And I put this, it's at the bottom, it's here. So there's 2 different study guides. So the exam for Thursday is 75 points. And part of it is on biotechnology, which is all the stuff that we're doing this week. So I think it's chapters 20 and 21. And then this one is the one I just added. So this has kind of what information from our lab project you should review. So it's stuff about C. elegans, stuff about RNAi, and then a little bit about our strain that we're working with, and then some of the techniques that we've done. So we've actually done a lot in the past so far, right? We've done model organism work, we've done molecular biology work, which is like plasmid purification, the gel electrophoresis sequencing, etc. So if you go through that stuff and you really have a solid understanding on those, you should be able to do well on that portion of the lab exam. And as a reminder, we're going to do 1 hour for the exam, and then you turn it in, and then you can work as a group. And if your score improves, it can raise your score, but if you did better as an individual, just keep your individual score. Okay, any questions on that?
Will it be the same format where you need a Scantron and then short answer?
Oh, that's a good question. Yeah, I guess there's multiple choice parts to it. So yeah, I guess we'll do a Scantron. I'll have extras to that. Yeah, and then there will be some short answer questions. So, just like I have here, how do you prepare an agarose gel to a certain percentage? So that might be a question of like, describe the steps that you would take to pour a 1.5% agarose gel. And you have to include, calculations for diluting the TBE, and then measuring out the agarose and how much gel green would you use, all of that. So it's kind of practical stuff from the lab. Okay, but if you read through this, you have questions, of course, let me know tomorrow. We can talk about any parts that you're unsure about. Okay, the other thing I wanted to mention is, I don't know, did the evaluation kit open today? Or did you see evaluations yet?
No.
Maybe it'll start tomorrow, but please take the time to fill that out. I will give some extra credit for doing that if you show that you've done the evaluations. For me, it's really useful to make improvements on a class, and I really honestly do listen to what you say. Aside from, you know, if you say like, there's too much information, like there are certain course outcomes that I have to cover. But, you know, if you have suggestions for like things that you liked doing or things that you didn't think were effective, let me know, because I always do listen to the feedback and I try to make changes every quarter. All right. So we're gonna go back to the biotechnology stuff. So yesterday we talked about, I think we left off with genomic libraries and cDNA libraries. So just to kind of review for a second, I want you to think with the person next to you, try to make a list of a couple of differences between those two. So what are the differences between cDNA libraries and genomic libraries? And then we'll kind of review this and get started and pick up where we left off.
Yes.
Well, cDNA is the complementary DNA, so it doesn't have any introns.
Oh yeah, that's true.
And then
Yeah,
genomic
has
is the introns, but it's also like spliced up differently.
Oh
Like
yeah,
in-
that's true.
I can't remember that.
I don't know.
Any thoughts?
Maybe that's because
He's not here yet.
I was like, oh.
You should try.
All right, so what is one difference between a cDNA library and a genomic library? So makes it introns. Okay, which one has introns? Yeah, so the genomic library, remember the way they get it? It's randomly chopping up the whole genome. So all the DNA is included in the genomic library, including introns. What's another difference that we talked about?
cDNA reflects the relative abundance of each mRNA.
Yeah, so we kind of talked about how they make it. So they're making it from mRNA, which means that whichever genes are being expressed, those are the ones that are going to have cDNA clones in there. So with genomic library, all of the parts of the genome would be represented. But for the cDNA, which parts of the genome are going to be found in a cDNA library? Would you find like intergenic spaces? Intergenic DNA? No. It's just exons. It's just the exon pieces that would be reflected. Okay, so let's say that you're going to do, back to our original thing, right? We said we were trying to make this human growth hormone. That was the challenge that we were talking about. So if you wanted to pick the DNA out of the library, how are you going to identify which piece has that human growth hormone in it? Well, one way to do that is actually to use a technique called PCR. So I hope that most people did learn about PCR, called polymerase chain reaction. It was created, it was developed, I should say, by this guy, Kary Mullis, but it was in the 80s that he developed this. So it's probably older than many of you. But anyway, the way that they do this is it's a way to automate amplification of DNA. So if you start with a tiny amount of DNA as a template, you can amplify it till you have millions of copies of that DNA. And so if you wanted to find, let's say that you want to find the human growth hormone gene within a cDNA library, you design primers that are specifically going to amplify human growth hormone. And then you could use PCR as the technique, and you can make millions of copies specifically just of your human growth hormone. So for number one, it says, specific primers can be used to amplify a target sequence from an uncloned genomic DNA or cDNA library. What uncloned means, I'm going to go back to that picture when we discussed how they make a cDNA library. So remember that they start with the RNA, mRNA, and then they reverse transcribe to get DNA from RNA. Then they make the second strand, so then it's double-stranded. They then clone it into a plasmid, like this. Now at one point you just have a whole bunch of plasmids, and you haven't yet put them into bacteria. When you have that collection of DNA before you get it into the E. coli, this would be called uncloned library. So you could stick this mixture of all these different plasmids in a tube with primers that are specific for one gene, and then it's only going to amplify that one copy. So that's typically how people will use a cDNA library to amplify out some gene of interest. Okay, so that would be one way to get human growth hormone. And for human growth hormone, would we want to use the genomic DNA library or the cDNA library if we want to ultimately have bacteria expressing? Yeah, we have to use cDNA because bacteria, they don't know what to do with the introns. The second point here is about, and this is kind of more, I would say this is probably older, like before we had genome sequences for all of these different organisms, but if you only knew the amino acid sequence, you could still actually identify that piece of DNA out of a library, because you can use degenerate primers, which is a mixture of primers. This is a little bit less applicable to what we're talking about with the human growth hormone thing. Okay, so let's talk about how polymerase chain reaction works. So for this, I said it was created in the early 80s. And the way that it works is that basically you stick a bunch of components in a tiny tube. It might actually have some PCR tubes in the back. I can show you how small. They're even smaller than those microcentrifuge tubes. You put the template DNA in there. You have to put primers. What else would you need to stick in the tube to get replication of a piece of DNA? So we need primers. What else? We need a polymerase. What is the polymerase going to use to make the strand? You need DNTPs. So actually, let me go to this slide. These are the requirements. If you want to make millions of copies, you have to have a polymerase. And I want to talk a little bit about TAC, that part. TAC stands for Thermus aquaticus. Is that, how do you spell aquaticus? So this is actually a name of a species of bacteria. The bacteria lives in thermal vents. So it lives at very high temperatures. So as you might imagine, its polymerase is heat stable. So this, I'll just say this is a thermal vent bacteria. So what scientists did is they realized, all right, if we're going to repeatedly heat and cool the DNA as a way to denature the strands and then renature them, we have to have a protein that can withstand that repeated heating. So they isolated the DNA polymerase from a bacteria that lives at very high temperatures. So its polymerase does not have any problems when it gets heated to 95 degrees. So that's the polymerase that we use for PCR. We have to give it the building blocks, so the DNTPs. And then for the primers, I'm going to talk a little bit more about how you design primers. But for this, we have a stretch of DNA that we want to amplify. And we need to have primers going in both directions. So if you have, let's say that this is the region where the human growth hormone is found, you need a primer that will give you a copy of the top strand. You also need a primer that will give you a copy of the bottom strand. So you're going to have to be able to design specific primers that flank whatever gene of interest you want to amplify. For the primers, I do want to make a note. So in the cell, what are primers usually made out of? What type of macromolecule? It's usually RNA, right? In the cell. In a polymerase chain reaction, you use DNA primers. The reason is, well, actually, can anybody guess why you would prefer to use DNA primers versus RNA primers? Yeah.
Is it because ribose is unstable compared
Yeah.
to DNA?
Yeah, exactly. So RNA is a lot more unstable. So it's better to work with DNA so we can chemically synthesize primers ourselves. And actually it used to be, back in the old days, this was somebody's entire PhD thesis was to be able to synthesize an oligonucleotide, but now we order them for $7. And they make them on a machine, really fast and easy to make any sequence you want. So you can basically tell the machine what order of nucleotides, and then it creates this short DNA oligonucleotide. So we use DNA for PCR. Then you need to throw in the template. So template can be a lot of different things. A template could be cells that you got from a prime seed that you've lysed open if you have like a small sample of DNA. It could be a plasmid. It could be one of the libraries, like a genomic DNA or cDNA library. Anything that you want to amplify from, you would have to stick into the tube. So for example, if we wanted to do PCR on your cells, we could, let's say that we wanted to do a test to see if you have a certain allele of, let's say a certain allele for sickle cell anemia or something, see if you carry that allele. We would take some of your cells, we could lyse them, and then that sample of DNA would be put into the tube with all of these reagents. The primers would be specific for that gene, the hemoglobin gene. So we could amplify it, sequence it, and see if maybe you have the sickle cell anemia allele and you're a carrier. Yeah.
So like for a COVID PCR test, would you be like trying to multiply the virus's DNA?
Yeah. So they're not using standard PCR because they're using a different type. It's a sort of a variation of PCR, but they are detecting the viral DNA.
By amplify, you mean like multiply it so you can actually test it?
Mhm.
Okay.
Yeah. So we'll look at how that happens. And then the other thing you throw into the mix is buffers and salts that help the polymerase to work. So you keep it at a good pH. The salts are used by the polymerase as a cofactor, so you have to throw those in as well. And then the last thing that you need is the machine that does this, and that is a thermal cycler. And I actually want to show you all. We have one right back here. This is a thermal cycler machine. So they're really simple. They have these tiny little wells. You can actually come and take a look at it at some point. The tubes, let me see if we have any back here. We use these tiny, actually these are different. Oh, PCR clean. These are PCR tubes that you can use. So you can basically, we have another rack of it. There's strips of them that are even smaller. But you stick those tubes in there. What the machine does, it can be programmed to heat and cool repeatedly. And that is how you actually get millions of copies. So let me show you the idea behind this. And then I have an animation that shows this. So the first step of PCR is to heat the two strands to separate them. So there's very common PCR programs that you use, so you should kind of be familiar with what is happening at each of these different steps. Step 1 is called denaturation, and that is heating it to 95 degrees. At that temperature, your two strands come apart, as you can see right here. And then each of those can serve as a template. So then, step 2, you cool it down so that the primer, the 2 primers can bind. And typically, I feel like I'm suffocating. Typically it's like 55 degrees for annealing. Okay, so that's when the primers will bind. At this temperature, the 2 strands don't come back together because it's easier for the primer to find the sequence than it is for the 2 strands to find each other. So primers will bind. And then the second step, or the third step I should say, is synthesis. So we also call this elongation. The elongation phase, that's usually 72 degrees C, because that's the temperature that the polymerase prefers for extension. So the polymerase extends. Okay, so then you get just one cut, now you double. So when you go from, in this first cycle, you go from 1 strand to 2 strands. You typically do these 3 steps at least 30 times. So 30 to 35 times. If you take that and do the math on it, you're going to get up to millions or billions of copies of that DNA by the end of the PCR cycle. So this is why this can be used for forensics. If you have a tiny sample of blood or a hair sample or something like that, you can get enough DNA and amplify it to then be able to analyze it. So let me show you the video of how this process works, because it kind of helps to see it animated, I think. Oh, and I don't have, there's no speaker cord here, so I'll just put the closed captioning on, and then hopefully I'll turn it up loud.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the prime sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Okay, so that kind of shows you what happens during PCR.
So you're literally just heating it and cooling it just repetitively.
Yes. Yeah, that was the genius, is that, wow, we can amplify DNA with a machine. We just need to heat and cool and put it in the polymerase. Yeah.
About how long does that take?
So usually, yeah, that's a good question. So every single cycle, this is like a 30-second, you do this for 30 seconds. The extension, or the cooling is usually 30 seconds. And then the extension depends on the length that you're trying to amplify, but they say like 1 minute per kb, 1 kb. Kb is 1,000 nucleotides. So usually if you're amplifying like a 500 nucleotide piece, you would do this for like 1 minute. So every cycle would take 2 minutes. So it takes like an hour and a half or so. If you do more cycles or whatever, it can be like 2 hours. Yeah. Okay. So that's kind of the idea behind this. You see that one question people sometimes ask is what stops it from working? So, for example, here when we add the primer, there's actually nothing that stops it. It's gonna keep elongating until you shift the temperature again. So there's really not something to stop it, but what happens over time is that, as you saw in the video, you start to have templates that only have, that start at a certain edge, and then if you amplify with the other primer on that template strand, you're gonna end up with a fragment that is flanked, and it's only the size. And after 30 cycles, that's mostly what you have. In the tube, there's a tiny bit of the original parent strand left, but if you have billions of copies, that's in the minority. You wouldn't even see that on the gel. You'll see the piece that you were amplifying. So the question is, how do we design the primers? So if you can think about this for a second, what do you need to know to be able to design those two primers?
Let's see.
You have to know the sequence, right? Yeah. So nowadays, now that we have sequence genomes, it's pretty easy to design primers. Even for like in C. elegans, we have the sequences for all these different genes. We can easily design primers. But the position is really important as well. So I want to teach you kind of general principles of primer design. With the molecular bioscience students, when they take the lab class, they design their own primers, and they run the experiment, and then they clone it in. So if you took these higher level classes, you might have an opportunity to actually do this. So primer design, you need a forward primer, which is this one, and a reverse primer, which is this one. The forward primer is also in this picture here. The forward primer is designed to make a copy of the top strand. So because it's going to make a copy of the top strand, it has to be complementary to the bottom strand, right? And importantly, you want the primers to go towards each other. So they flank the sequence that you want. They also are building towards each other. So if we have this primer right here, which way is it going to be building off of? Which direction? This way, right? 5' to 3'. This one, 5' to 3' is going to make a copy of the bottom strand. So it has to be complementary to the top strand. It's going to build a copy of the bottom strand. So that's kind of, the placement is really important. It can be very confusing for students. So I actually have on our ICA, you get to practice, okay, how would you design it if you're given the flanking sequences? So in a second, we'll take, and you'll have a chance to try to figure out where would you want to then design it. The other things that you need to take into consideration, how long they are. Usually the ideal length is between 18 and 30 nucleotides long. The sequence is completely dependent on what the strand that it's complementary to is, right? So one thing I want to point out, if this is your forward primer, why is this bouncing? I don't know if that machine is just shaking or what. Or am I imagining it? Okay, so this sequence, it's complementary to the bottom strand, but it would be the same sequence as what the top strand is, right? So when you're designing a forward primer, you can actually use the exact sequence that is on the top strand. You need to find a region that is 18 to 30 nucleotides long. Other guidelines, you want to make sure that the GC content is around 50%. So they give you a range of 40 to 60. Any ideas of why you would want to keep GC content kind of limited? What if G's and C's, how many bonds do they have? 3. So they have it takes more heat to separate them. So you don't want it to be high in GC content. It's actually hard to get those to come apart. That can affect the melting temperature. You don't really need to know, how to figure out the melting temperature. But if you roughly have 50% GC content, usually the melting temperature is good enough for getting annealing of the primer in a specific way to its target. Now, how are you going to figure out if you have 50% G's and C's? Just count. So if you had 30 nucleotides, you want to try to aim for about 15 G's or C's in your sequence. Now, obviously you have a range 40 to 60, so you try to keep it, just count up how many G's and C's are in this stretch, and is it within that region of 50%. Then the last point is you don't want the primers to be able to fold up and form a structure, a secondary structure, and that's through self-annealing. So like if there was some parts of the primer that were self-complementary, you might be able to get like a hairpin formed or something like that. That's gonna be competing, this structure will be competing with the binding on the actual target sequence, so you don't really want that. And how do you know that that's gonna happen? There's actually a lot of tools that people have developed where you put in a sequence and it tells you whether or not there are secondary structures. So you don't, in this class, don't worry about it, but just really, know that you want to check for that if you were actually going to order some. And primer dimers is when your two primers, forward and reverse, have some complementary regions so they come together. And of course, that's also going to compete with those primers binding on the target. So you want to avoid that as well. Okay, so I want to show you this. PCR seems straightforward, right? But actually, it's in a lot of labs, it's a very touchy thing, and sometimes you have no idea why it didn't work. The primers should in theory amplify, but they don't, or they have off targets that you didn't expect. And so this is just showing a shrine people set up to the PCR machines. And I have a video that was developed by Biorad. It's kind of funny. This can help you to actually remember the steps of PCR. So let me show you this video. Okay, yeah, this was developed as an ad. Oh.
There was a time when to amplify DNA, you had to grow tons and tons of tiny cells. And don't give again and talk to Carrie Morris. Said you can amplify in vitro just as well. Just speak your tablet with a pulver and some primer. Nucleotides and polymerases too. Neighboring, annealing, and expanding. Well, it's amazing what heating and cooling and heating will do. PCR, when you need to detect mutation. PCR, when you need to read the blood. PCR, when you need to find out who the daddy is. PCR, when you need to solve a crime. PCR, when you need to detect mutation. PCR, when you need to read the blood. PCR, when you need to find out who the daddy is. PCR, when you need to solve a crime.
All the scientists out there doing PCR, Biolab salutes you with the all-new 1000.
Okay, so that was developed as an ad. I think it's funny, I don't know. I have a weird sense of humor. But anyway, so that's kind of the basics for